Multiple sclerosis in 2018 is very different than it was in 1993 when we had our first injectable interferon treatment. We now have many compounds that suppress disease activity much more effectively than when we started this 2 decades ago. But there's still a significant amount of unmet needs here. We have metrics that we call NEDA, no evidence of disease activity, and that metric looks at clinical attacks, disability progression, and MRI. But that's not the whole story of MS, because there are pieces that are not measured, and there's this piece called progression that occurs independent of everything that we have to use to treat MS. So one of the problems is you look at all the drugs that we have available, even the most potent of them, and we're only getting into the 40% NEDA range, 40, 45, 48%, whatever it is. But you're not getting to 100%. And that's not even taking into consideration other metrics that we have not even added to that metric, such as cognition, huge issues, fatigue, bladder issues, spasticity, I mean, you name it. There's a lot of disease activity that kind of goes undiagnosed or untreated because we're focused in on those metrics that we can easily see, but all these other ones are going on. And so in that other 50% of the NEDA that's not met, there's that, and then there's the piece called we've suppressed the MRI, we've suppressed their clinical attacks. We're slowing the rate of progression, but we're not stopping progression. People still get worse. So this phase of progressive MS is a huge unmet need. We need better biomarkers, no question about it. We need our abilities to look at individual patients and say, okay, so this is somebody, like I may have a blood test that I could get for somebody and say, here's somebody who's not going to do well in the next 5 to 10 years by this blood test. I got to put you on this treatment because this is going to be the right drug for you. I may have somebody who doesn't have that marker and says, you know, this is somebody who's going to do okay. We could do other treatments. But we don't have those biomarkers. Right now, there's some ideas that maybe we'll have something called neurofilament light chain that may potentially be a marker that might help us in this world. But we need better biomarkers or tests to look at cognitive function, to look at things on MRI that we can't see so easily right now on our conventional imaging. There are new metrics that are being applied. Even tests related to the thickness of the retina with those CT scans that may give us the ability to deal with this slow progressive phase of the disease, and maybe we can start targeting these biologic events. Can we remyelinate? Can we protect the nervous system in a way that you don't get loss of neurons and loss of function? So that's ultimately what we need to get to. We're not there yet. We've made tremendous progress over the last 2 decades, but we have a lot more work to do.
Moving forward, MS continues to be an extremely exciting and interesting therapeutic area. We've gotten a great handle on relapsing remitting disease. Not perfect, but really control it very well. We've opened the door for treating progressive disease with one agent with modest effects, but available. Another agent going through the approval process now with a modest effect on secondary progressive MS. Some agents and strategies in the pipeline to hopefully improve upon that. And there's a number of studies going on. So near-term excitement is better treatment of progressive disease. And it's interesting in MS, the clinical trials have been directing the science. You know, we learned about the disease through successful clinical trials. We learned about it through basic science and animal research as well, and the two kind of come together. But in MS, we've really learned a lot by having a successful clinical trial and then saying, well, why did that happen? And sometimes why it happened is different than what we hypothesized when we started using the drug. And so that area, progressive area, is moving along nicely. And then we have agents in the pipeline now for repairing the damaged nervous system, which is the most exciting thing that we're doing, you know, short of a cure.